All right, in the previous lesson we looked at the transmission of disease and the first line of defense being a non-specific form of defense, and today we're going to start part one of two lessons on the second line of defense. This is still a non-specific form of defense, but now we're looking inside the body. So today we'll look at macrophages and inflammation. So second line of defense, as we've mentioned, okay, is where pathogens are going to now enter the body. Okay, so the first line of defense isn't perfect. Pathogens do make their way past those defense mechanisms. Now, once they've made it inside the body, we have several non-specific defense mechanisms, and this is going to prevent these pathogens from becoming established within the body. Okay, so some examples: macrophages, inflammation in this lesson, and then in part two, we'll look at fever and the lymphatic system very shortly. So, phagocytes. What are they? Well, number one, we have a general definition here, leukocyte. This is the name given to white blood cells. We learned this in year 11. A phagocyte is a specific kind of leukocyte which is capable of a process known as phagocytosis. Now, the cells that commonly undergo phagocytosis are macrophages. And a macrophage is a phagocyte that engulfs and destroys pathogens. They're found within the blood and lymph nodes, and they leave the bloodstream through capillaries where they travel to a particular site of infection. So what they're going to do is they're going to sample the cell material, and if they find that that cell is non-self, they're going to ingest it and they're going to destroy it and break it down. We will look at this process in great detail today. So, number one, what's our first step? These phagocytic cells are going to sample the cell material. They're going to determine whether it's self, aka coming from the body, or non-self coming from outside the body. If it's found to be non-self, they're going to ingest it via a process known as phagocytosis. Okay. Now, we can see this taking place up the top here. We've got all of these microbes. They're found to be non-self, and the cell wall begins to pinch around them. This forms what I'll highlight in green here. We've got step 3, a vesicle forming. This is our vesicle forming. It's formed around the microbe or around the pathogen itself. Now, what comes and binds, if I highlight it in blue, is a lysosome. A lysosome, we spoke about this in the first line of defense. A lysosome is a organelle within the cell which contains digestive enzymes which binds to the vesicle, leading to the digestion of the microbes themselves. And we can see, if I highlight in pink here, we've gone from our microbes here, and then we've ended up with fragments everywhere here. Finally, the phagocyte, the macrophage itself, doesn't want this particular broken down material within its cell, and so it gets rid of it via another process, and I'll get rid of my pink highlighting so we can use that here, known as exocytosis. So what I've done is I've broken them down here with a slightly different diagram to represent. I'd encourage you to have a look here, to pause it, and to go through each step. The only change that I would make, especially at a year 12 waste level, is instead of the word phagocyte, we're going to use macrophage. Okay, just to be more specific and more detailed. So macrophage and macrophage. So, moving on down, another representation, just have a read through, not to attach to this one, before we get to inflammation. Okay, so we've looked at phagocytosis. We've seen that these cells rock up when they detect a non-self cell, they'll eat and ingest the non-self cell, break it down. But what we can also have in the second line of defense is this process of inflammation, where there's a localized response to damaged tissue. Okay. And what happens in this process is we're going to stop pathogens from spreading, we're going to destroy pathogens, we're going to get rid of damaged tissue, and we're going to begin the repair of that tissue as well. So things to note, you've probably heard of some examples of inflammation before. Anything ending in itis is generally inflammation. So appendicitis or tonsillitis, all of these things are inflammation of particular organs around the body. So, couple of key steps here. Let's go through them one at a time. Number one, the first step of inflammation is there must be damage to tissue. What kind? Either mechanical damage or chemical damage. What happens here is this stimulates mast cells, okay? These are special leukocytes, and what they do is they release two things. They release histamines and they release heparin, okay? They also release other chemicals which attach to phagocytes themselves to identify them, okay? So step one, damage occurs to the tissue, either mechanical or chemical, triggering mast cells, which are special kinds of white blood cells, to release histamine, heparin, and other chemicals that bind to phagocytes. The histamine is the really important bit for us. That's the one that we really care about. Heparin, it's good to mention, but it's not our key idea here. So histamine, what does it do? Two things. Number one, increase the permeability of blood vessels to allow more plasma to escape the tissue. And number two, vasodilation. We're going to increase the blood flow to the area. Why are we trying to do this? Well, it's so that we can bring more macrophages to the area itself. Heparin, because we've increased the permeability of the blood vessels in the local area, we want to prevent it from clotting. Okay, we want to prevent clotting within the local area, and so heparin prevents that clotting process from taking place. So when we're having a look, our final part here is our phagocytes, due to the action of histamine, have been able to not only arrive at the site of infection, so we can see all of these green microbes on this image here, but they're then surrounded by these phagocytes which have been able to escape from nearby blood vessels, surround them, and then they're going to consume all of these microbes or pathogens or cell debris, depending on the kind of damage. Now, from here, pus will form. Not always, but most commonly. And pus is where tissue fluid, okay, dead phagocytes filled with all of those nasty microbes and bits of debris, fail to clear and gradually drain and clear the site, okay? And what's going to happen as the pus gradually comes down, new cells are also produced by mitosis to help us repair that damaged tissue. Inflammation, again, is not a process only where we're protecting the body, we're also trying to repair the body now. And in this process, we end up with 4 key symptoms. Okay? So the 4 symptoms that are commonly associated with inflammation are firstly redness, and the reason that we get redness is increased blood flow to the area. Secondly, heat. This is because the heat of our body is most commonly carried in our blood, and if we're increasing how much blood is coming to a particular area, it's going to feel slightly warmer. Third, swelling. Swelling is where we're going to get that increased fluid movement to the intracellular space, and so it's going to slightly swell due to that excess fluid. And finally, pain. Okay, so pain is going to be triggered by nociceptors, and that's a result of either the pressure from all of the swelling or maybe even from toxins which have triggered those nociceptors. So this here is a final representation showing quite a few of the different options and steps which are going on with inflammation. You might like to pause and have a read through.